So, genetic variation, it is necessary for crop improvement in agriculture. So, what all different ways in which crop yields are improved or their traits are improved? Hybridization. Now, what is hybridization? The process of combining different varieties of the same plant. I will be talking about plant hybridization of the same plant, such that then you select for or the hybrid lines which will have the combination of the good traits of both the varieties. So, it although includes, it is again a random process. So, this will include a selection of the cell lines which will have all the positive traits which have come together. Then mutagenesis, DNA may be modified either naturally or artificially by a number of physical and chemical agents resulting in mutations, and then again you do a screening and selection. Then polyploidy, you apply certain agents which can result in change in the ploidy. In plants it is known. So, which may lead to improvement in the traits desired. Now, plant cell cultivation in vitro generates a considerable amount. Now, we know that under in vitro conditions we have heard about soma clonal variation, which itself can be used to generate different cell lines of different genetic makeup, because this can lead to polyploidy, change in the karyotypic nature of the cell. So, somatic hybridization is what? It is a well known technique, we have learnt about it. So, somatic hybridization, in which somatic cells are fused together such that the cytoplasm as well as the nucleus will get fused, and therefore, you will end up in a single cell which will have club of both the varieties of cells. So, generally it is observed that these hybrid cells which come out, they will be more vigorous in nature. For crop improvements, you will see that hybridization of plants is very commonly used. So, seedless fruits, like example, I do not know whether you have heard or seen seedless papaya or very bigger size fruits. So, these are all because of hybridization. So, generally it is deliberately pollination is done, mixing is done, such that the hybrids are generally observed to be more vigorous in nature in terms of their growth rates or in terms of yields. Now, the plants which are obtained through genetic engineering contain a gene usually from an unrelated. So, when we say transgene integration, it means that a gene which is not present in the plant but has been transformed in the plant chromosome. So, such genes are called transgenes, and the plants containing the transgenes are called transgenic plants. Genetic transformation can be defined as the transfer of foreign genes isolated from plants, virus, bacteria into new genetic background. The first step in gene transfer is to select the cells that are capable of giving rise to whole transformed cells. So, we will see what are the different ways in which the selection is done and what are the different ways in which the transformation of plant cells is done. So, what is a gene construct? When we are transforming plant cells, this will consist of, this is common. So, gene construct for transgene integration will contain different assembly of DNA. Generally, agrobacterium is used as a tool to transfer this genetic material. There are other techniques also which we will be talking about. So, the genetic whole transgene which is transferred will have these many sections as numbered here. So, the construct will contain a promoter, will contain an enhancer or silencer, transcriptional start, leader sequence, initiation codon, exons, introns, stop codon, untranslated regions and poly A tail. Have you heard about these terms? You have heard. So, promoter is required for, promoter is used for, be loud, RNA polymerase, the transcription of the gene. Then enhancer and silencer, they regulate something. Right. So, they regulate, as he said, the promoter activity. When it is an enhancer, so it is enhancing the promoter. When it is a silencer, it will silence the promoter. This can have certain sequence, or this can lead to selective induction of the promoter by either exogenous addition of certain agents. So, this is how it may lead to regulation of the promoter activity. Then transcriptional start, leader sequence. These are the regions of mRNA for the regulation of translation part. Then at the ribosome level, because once the mRNA is formed, it will attach to the ribosome for translation. So, there this leader sequence regulates that. Then initiation codon, exons, introns. So, what is responsible, where your desired gene is, which part of the gene is responsible for expression, introns or exons? Exons. So, there is RNA splicing which removes introns. Then, but regulatory role has been found of the intronic regions as well. Then stop codon, which is the transcriptional stop. It is followed by untranslated region. So, there is 5' untranslated region, 3' untranslated region. 3' untranslated region will also have your poly A tail, which is required for stability of the mRNA for further processing. So, the promoter, it provides the site for binding of RNA polymerase and is involved in transcription initiation. Then it is required for an efficient expression in plant cells. So, generally the promoter which are used are either can be inducible promoters or can be constitutive promoters. What is inducible promoter? Which means the promoter activity is regulated. In the presence of certain chemical, the promoter will be active. Then constitutive promoter, irrespective of the conditions, it is uniformly all the time active. So, CAM-MV35S promoter, have you heard about CAM-MV, which is a very widely used cauliflower mosaic virus promoter for genetic transformation in plant cells. Now, is this a constitutive promoter or an inducible promoter? This particular promoter is responsible for the entire gene transcription of mosaic virus. It is a constitutive promoter and very high activity. That is why, and it is well known to be infecting the plant cells. So, this particular promoter, being a constitutive promoter, a strong promoter, is used for genetic transformations in plant cells. Among all the promoters known, CAM-MV35S promoter is very commonly employed in transformation experiments. The promoter of the 35S RNA is a very strong constitutive promoter that leads to the transcription of the whole CAM-MV35S. So, it is well known for its use in plant transformation. A suitable enhancer sequence is also required to control the activity of the promoter. Now, so why do you think this enhancer sequence, how is it useful? If suppose you want to express it at a particular developmental stage, or you want an expression in a particular tissue, so then you need to regulate the activity of the promoter. So, that can be done using your enhancer or your what else? Silencer sequence. The enhancer is defined as the DNA sequence which enhances the activity of the promoter, and the DNA sequence suppressing the activity of the promoter is known as silencer. So, how do we recover transgene cell lines? Suppose we have done transformation using this construct. So, how do you find out that which particular cell line is transformed or not? How do you select? You would be needing a marker system. Now, what is a marker system? Antibiotic. So, generally used is antibiotic resistance. So, it is a selective marker system. Now, in which, what does it mean? Only when it is expressed because it is present in the transgene which is integrated into the plant chromosome, it will express in the plant chromosome, then only it will be able to show antibiotic resistance. Now, even in antibiotic resistance, generally the antibiotic, you would like to choose an antibiotic resistance against an antibiotic to which the plant cells are highly sensitive. So, what kind of antibiotics? Generally hygromycin is used because plant cells, kanamycin we have heard in bacterial transformations, but plant cells are found to be highly sensitive to hygromycin because of their uptake and their reach, availability to plant cells and affect them. So, hygromycin system is used as a reporter system or as a marker system. So, selective agents differ in their toxicity to different plant species. The different developmental states of the plant cells or tissues give different response to the selectable marker. It is necessary to use the correct concentration of the selective agent. Then herbicides are generally more toxic to plant cells than antibiotics. Now, callus and cell cultures are better systems than organized explants to achieve transformations and for the selection of transformed cells. Now, reporter system, how is it different from your marker system? Generally, your GUS, your GFP you must have heard, isn't it? Why is it used? So, try to connect it with saying the word reporter gene.
You want to know what levels of protein are being expressed, so we can correlate it with.
It can be quantified as well as qualitative in nature. So, if it is qualitative in nature, it will tell you what? Expressed or not. So, generally these systems in plant cells, you will hear GUS assay is very frequently used, which is glucuronidase assay or luciferase assay. So, in which blue fluorescence or blue colored product comes on the cells, which are once they are exposed to X-gluc substrate. So, this particular enzyme, if it is expressed properly in the transgenic cell lines, then will be able to break down the substrate X-gluc into product which is blue color, and you can visualize that, and then this becomes an indication of successful transgene integration. Now, the genes need to be tagged with another genes which are called as reporter genes. Now, I was saying that you can confirm whether it is a successful transformation or not using reporter systems. Now, the other thing, how it can be, what else can be the use of reporter system? It can also be used to check promoter activity, or the activity is, the promoter is active or not. So, then if you use the same promoter which you have used for your desired transgene integration for this particular GUS or luciferase activity, and if that gets expressed, then there is a high chance that the promoter is intact, the promoter activity is intact, is able to show good activity in this particular plant species. So, a reporter gene is a coding unit whose product is easily assessed, like GUS, whose product can be easily detectable. So, it is called a histochemical assay. So, this gene may be connected to any promoter of interest so that the expression of the gene can be used to assay promoter function. That is, it describes the transfer and activity of the promoter. Two genes that are widely used is beta glucuronidase and the other is luciferase. The coding regions of these non-plant genes, so these are transgenes, non-plant genes have been fused to plant promoters and polyadenylation sequences, which means you need a 5' UTR and a 3' UTR. So, 5' UTR, you are using the same promoter which you are using for your desired gene, and 3' UTR for the entire construct to happen, your GUS or beta glucuronidase or your luciferase enzyme for expression. So, plasmid mediated gene transfer, how many different types of plasmids are used for gene transfer? These are conjugative plasmids or transmissible plasmids. The others are non-conjugative plasmids, which are also called as non-transmissible plasmids. So, what are plasmids? Self-replicating extrachromosomal DNA maintained as independent molecules. Generally, they are found in bacterial. So, plasmid DNA can be circular or linear in nature. The size of the plasmids varies from 1.5 kb to 1500 kb. So, conjugative plasmids, what are these? They can mediate their own transfer between bacteria by the process of conjugation. So, there is appendages, which are formed, and then the two cells will come together so as to facilitate the transfer of the genetic material. So, then conjugative plasmids can mediate their own transfer between bacteria by the process of conjugation. Then non-conjugative plasmids are not self-transmissible. So, generally when we are using non-conjugative plasmids, their transfer is done with the help of conjugative plasmids. Now, plasmids can be classified also in terms of lower and high copy numbers. Low copy numbers when it is nearly 1 to 4 copies. High copy numbers greater than 100. So, conjugative plasmids are large with stringent control of DNA replication and are present in low copy numbers. Non-conjugative plasmids are small, high copy number and have relaxed control of DNA replication. So, if suppose you are working for cloning, multiplication, what kind of plasmid you will prefer? Non-conjugative plasmids. Large number of copies are produced because of the relaxed control of DNA replication. High copy number plasmids are useful for expression of cloned genes to higher yield. So, when I say binary vector, these plasmids can get expressed in more than one different types of organisms. Like when in plant transformations, binary vectors like P. cambias, they can get expressed in E. coli as well as multiply in E. coli as well as in Agrobacterium. When such plasmids are used as vectors, they completely utilize the host metabolism. So, what is the demerit? They will utilize the host metabolism if they are expressed in a organism. Each plasmid has an origin of replication. Can you make out that why do we say it is a metabolic burden then, any kind of transgene integration? Each plasmid has an origin of replication. Plasmid DNA in bacteria may be 0.1 to 5% of the total DNA. Now, gene transfer methods, one is indirect gene transfer and the other is direct gene transfer. We will take up the direct gene transfers later. The indirect gene transfer methods are the most frequently used in transgenes integrations into plant cells. Indirect gene transfer, this is vector mediated gene transfer. Vectors are DNA carriers into which foreign DNA or genes of interest are inserted to make a recombinant DNA. Now, what are the different kinds? Cloning vectors and expression vectors. Cloning vectors which are required for if you want to multiply, want larger number of copies. Expression vectors, these expression vectors are used when you want to express the transgene into the plant, used for expression of the clone gene to produce the product. Now, most vectors carry what? We have already studied this, a reporter gene which allows recognition, example antibiotic resistance. So, plasmid mediated gene transfer. Plasmids are most commonly used vectors in gene cloning. What are the different types of plasmids which are used? So, different types of plasmids which are used are, you will come across TI plasmids or RI plasmids. TI plasmid is tumor inducing plasmids or root inducing plasmids. Now, some of these once disarmed, now I will come to certain diseases in nature because of bacteria which is Agrobacterium which contain these plasmids. Now, these plasmids will have set number of genes. Now, why do you think a bacteria infects the plant? Should be a purpose behind this. So it contains certain genes which cannot be expressed when the plasmid is present in the bacteria, but as it gets integrated into the plant, they will express these genes, and these genes lead to opine production. These opines are carbon and nitrogen sources for the bacteria. So now in this course, the portion which gets integrated will have certain oncogenic genes. Now the plant is allowing this infection to happen, so it has certain auxin and cytokinin genes as well which get integrated, which will cause some biochemical changes to happen, leading to neoplastic growth at the point of infection. Now morphogenetic events can either lead to rapid multiplication of cells leading to crown gall formation, or organogenic or adventitious root formations which are called as hairy roots. Now this itself, therefore, these agrobacterium and the plasmid containing it has then been modified or disarmed. Disarmed means removing of these oncogenic genes, which means your auxins and cytokinin genes, and putting an MCS there, multiple cloning site, with different restriction sites, so that the desired gene can be integrated. Now this whole cassette is included in that tDNA region, that portion of the construct which gets integrated into the plant chromosome. This is called as tDNA. Now this portion contains a right border and a left border. These are direct repeat sequences, 25 base pairs around long. So there is right border and left border. Then the entire outside this tDNA also there are different regions of the Ti plasmid or the Ri plasmid, the genes which are responsible for the breaking and the multiplication of this tDNA, processing of this tDNA, then transfer of this tDNA into the plant cell, protecting there till it reaches the chromosome of the plant cells and gets integrated. So there are virulence genes, then there are other genes which are required in the entire packaging of the tDNA. So it is not alone tDNA, but it is tDNA with other proteins which are involved in this entire process of movement till the plant chromosome, even with its interaction with the plant, with its interaction with the plant proteins, thereby facilitating the integration in the plant chromosome.